have today Jan Hengstler. I invited him because I cannot see you with this. I will try not to connect with the computer to have you on video. But he's a professor, a W3 professor at the Leibniz Research Center in Dortmund, and I invited him because he very recently he just published a outstanding paper in Journal of Hepatology, associating the bile acids and acetaminophen metabolism. So I think he's going to show us very nice pictures today with imaging and hepatotoxicity. The title of his talk is Functional Imaging of Hepatotoxicity, the Breach of the Bile-Blood Barrier. So Jan, sorry for the delay, and you can start whenever you want.
So thank you very much for your introduction and the possibility to discuss some results of functional imaging with a focus on the bile-blood barrier, and which is breached by after acetaminophen exposure. And let me start with just a second. It's working again. Let me start with some basics with which most of you, of course, will already be familiar, but these structures will appear again and again now in the imaging. So the liver consists of these smallest units, the lobules, which all have a similar design. It receives blood from branches of the portal vein and oxygen-rich blood, which then flows along sinusoids, along such sheets of hepatocytes, and drains into the central vein, and during this process is in contact with these sheets of hepatocytes. And the hepatocytes secrete bile into very thin bile canaliculi, which are connected to bile ducts and which drain to the intestine. This is the structure in detail. These sheets of hepatocytes with the bile canaliculi, they are formed by the membrane of the hepatocyte, so the apical part of the hepatocyte membrane. Here are the endothelial cells which line the sinusoids. They have pores, not shown here, so that the blood is always in contact with the hepatocytes. Between hepatocytes and endothelial cells are the stellate cells, cells which are contractile and can regulate the diameter of the sinusoid. They also store vitamin A and play an important role in liver fibrosis. Another cell type which we will constantly see is the Kupfer cell, the tissue macrophage. And while these are schedules from textbooks, it is of course clear that in reality bile canaliculi are not such straight channels. They are shown here in this figure. In reality, they are a complex network which is connected via these channels, also named hearing channels, to the interlobular bile ducts. And also the, by the way, can you see my cursor?
Yes.
Thank you. These sinusoids are also not straight. They also form a network, and both networks, the canalicular and the capillary network, are intertwined in a complex manner. So these are the basic structures which we will see again and again. And one of the imaging techniques I will repeatedly use in this presentation is two-photon microscopy of intact livers of anesthetized living mice. So what you need is an anesthetized mouse with a median incision. So there is a small, about 5 millimeter incision, so that the liver is in contact to this imaging glass. And then you need a specific microscopy setup. The first excitation by 2 photons. So these are low energy photons, infrared, because the lower the energy, the deeper they penetrate into tissue. And at least two photons meet in the imaging plane and reach the energy required for emission. Then what is needed is a long distance objective and a very sensitive detector. And then you can observe processes in the living liver, which helped us a lot. We should also be aware that we image relatively at the surface of, so only 5 to 75 micrometers below the liver capsule. We can go deeper. We can even image 150 or 200 micrometers below, but there we lose resolution. And since the architecture of the liver is so repetitive, one learns already a lot if one images relatively close below the surface. And what is also needed are reporter mice. But today, reporter mice which show fluorescence of all relevant cell types of the liver are commercially available. So just to give you some insight, this is a type 2 reporter mice, which visualizes the endothelial cells. You can see them here in green. Or here the stellate cells. Or here the Kupfer cells. Here one sees nicely how they are moving, and with their cell protrusions, they are capturing bacterial fragments to clean or to clear the blood from LPS and other particles. And this will be the main cell type to focus on today, the hepatocytes. So usually we visualize them by injecting a mitochondrial vital dye, TMRE, into the tail vein of the mice. Then they appear red, and you can even see the individual mitochondria. But this is only to visualize the hepatocyte cytoplasm, so that you see these sheets of hepatocytes. And in between are the sinusoids, so the capillaries. And the standard procedure is that we now inject into the tail vein a fluorophore-coupled compound, for example, a bile acid analog. We frequently use CLF, which is commercially available. And what I will show now is how, after tail vein injection, the green CLF appears in the sinusoid, then goes to the distal space, into the hepatocyte, and from there is secreted into the biocanalicus. Start the video. Sinusoid, here is the distal space. Now it is transported into the cytoplasm of the hepatocyte. And if you focus on this structure here, this is a biocanalicus here. And within a certain time period, the blood is cleared, and also the hepatocytes are cleared, and all of the bile acid analog is in the canaliculus, from where it moves on its journey to larger. This is here we have now seen the secretion in the canaliculi. This network is connected to the interlocular ducts, and they are connected to larger ducts, and finally trained to the intestine. And imaging of the interlocular ducts is also possible, of the larger ducts, but a bit more challenging because we have to go deeper. For our focus today is on this canalicular network. And the first example I will show is the breach of the bile-blood barrier induced by paracetamol. And I would like to give a summary in the beginning, then you can more easily focus on the experiments and discuss and criticize them. So these are two hepatocytes with a biocanalicus in between. The first observation, always when we deplete glutathione, this can be by paracetamol, but also by BSO, or other compounds which deplete glutathione, what we always see is then the bicanaliculus becomes wider. And then in the presence of high levels of oxidative stress, which is induced by paracetamol, the bicanaliculus does not only become wider, but it becomes leaky. So the tight junctions, which normally seal the canaliculus, are no longer functional, and it opens to the distal space, so to the extracellular space. Then the highly concentrated bile from the canaliculus leaks out in the distal space, but a high fraction of it is taken up again into the cytoplasm of the hepatocytes, is excreted or is secreted again to the canaliculus, leaks again. And this causes some futile cycling, which leads to higher concentrations of bile acids in the hepatocytes. And I will show that they increase beyond a threshold which causes cytotoxicity. So if we interrupt this reuptake, and with this, the futile cycling, APAP-induced hepatotoxicity can be strongly ameliorated. But now to the experiment which led to this concept. First, a few basics. Of course, a lot of work has been done about paracetamol. By the way, it was already discovered in 1877. It is known to be metabolically activated by cytochrome P450 enzymes to a reactive metabolite, NAPK, which binds to glutathione and to proteins, activates Jun kinase, which then translocates to mitochondria, which induces even more oxidative stress. And this can induce cell death. So cell death occurs in the center of liver lobules. And if there is a single intoxication, it's easily regenerated. So relatively large pericental necrosis can regenerate within a relatively short period of about 6 days in mice.
To brechen?
Yes, welcome. And so these pericental necrosis, you can also see here as white spots. So these white spots correspond to this necrosis. So why is the necrosis in the center of the lobules? This is because cytochrome P450 enzymes, for example, SYP2E1, are expressed in the central region of the lobule. And here you can see then this necrotic region. Since long, even before 2014, there were hypotheses if bile acids contribute to paracetamol hepatotoxicity. It is known that after intoxication, also bile acids increase in the blood of patients after overdose. And there is one hypothesis stating that this may be a secondary consequence of tissue damage. So that's quite often that upon tissue damage, the bile acids increase because most bile acids are in the intestine, reabsorbed via the portal vein. And if the liver loses its excretion capacity, the concentration of bile acids would increase. So this would be trivial. There was also a hypothesis that, bile acids, their increase is an early initiating event due to a breach of the bile-blood barrier. This was initially an outsider hypothesis, but this is for what we found evidence that it is correct. Yeah. And so what this hypothesis means is that the bicanaliculus, which is shown here, that this becomes leaky and releases bile acids at a very early time point after intoxication. This is in humans difficult to study because the data one usually obtains from patients is like this. So this is from an intensive care unit close to us in Essen. And the one there gets a very heterogeneous situation. So for example, patient 1 has high bile acids and low liver enzymes, but another patient shows the opposite situation. So high liver enzyme, or for example, here is high liver enzyme, low bile acid. And some patients show both high liver enzyme and high bile acid. So difficult to interpret. And I think this is because usually the exact time point of APAP overdose, or the interval between overdose and blood sampling, is not known. And here, Hartmut Jeschke and Stephen Curry helped us. So this was really an unusual situation that in the case of an 18-year-old woman who ingested a very high dose of APAP, that she sought care already 70 minutes after ingestion, and this period was very well documented. The APAP blood concentrations were 2.2 and 3.7 millimolar about 2 and 4 hours after ingestion. So very high, and it was possible to time-dependently sample blood. And what was quite striking is that the bile acids, here the blue dots, they increased before the liver enzymes increased. So this was the first evidence that an increase of bile acids may be an early key event. And to study this under controlled conditions, I used mice with a hepatotoxic but not lethal overdose, 300 milligram per kilogram paracetamol. And we took blood from 3 positions: from the heart blood, so mixed venous blood, from the hepatic vein, so the liver outflow, and the portal vein. And let's first focus on heart blood. That's the red bar, heart blood, and you see that there is a transient increase 2 hours after overdose, and then it decreases again. Hepatic vein, similar situation. Here, the gray bar, and also this transient increase, so the bile acids come indeed from the liver. The portal vein, this is a different situation. This reflects which bile acids are reabsorbed from the intestine. And what is important, this peak of bile acids in the blood is before the liver enzyme, here ALT, increases. And we see also from staining of the tissue, for example, in the tunnel assay, but this is only one readout. So we see that dead cells appear only 4 and even more 8 hours after intoxication. So tissue damage occurs after this transient increase of bile acids also in the mice. And this motivated us to go a bit deeper and now apply this intravital 2-photon imaging. This was one of the first observations which triggered us to think that something special happens. So this video begins 1 hour and 25 minutes after an APAP overdose. In red, you see TMRE, so which is mitochondrial activity. And green is CLF, the bile salt analog. And perhaps when I start now the video, focus on this cell. You see that here a protrusion forms. It gets larger, seems to be leaky, then the remaining cell becomes also green, and these protrusions are observed in many hepatocytes. And it took us more than 2 years to understand what happens here. But the first step in understanding such videos is always a systematic image analysis. And There we use a system which by artificial intelligence assigns every pixel of such a video to a certain cell structure. So does it belong to a bicanaliculus, to the cell, or to the sinusoid? And if this is now a control, a healthy mouse, and if I start this video, so you see where the CLF goes, the image, and simultaneously the intensity in the image analysis. And these are the bicanaliculi, which increase and decrease. But very important, red is the CLF signal in the sinusoidal blood. It peaks, of course, immediately because it's an intravenous bolus injection, but then rapidly decreases. So between 5 and 10 minutes, you go back to background levels. Black is the CLF signal in the cell, which also increases and rapidly decreases. So perhaps remember how fast the blood clears in the normal mice, 5 to 10 minutes. Now we come to the situation, APAP overdose, again starting 1 hour and 25 minutes after the intoxication. Again, the bicanaliculi here, but when you see the red lines in the blood, it stays high much longer. So this means that the clearance from the blood and also the clearance from the hepatocytes is much slower after APAP intoxication. By the way, the upper line is always the pericentral region where there is more APAP-induced damage, and the dotted line is in the periportal region. So when we quantify these labs, there is a systematic increase over a certain time period quite early after intoxication, and also the total lab number increases. And when we look at the diameter of the bicanaliculi, they become wider much earlier, very early. And by the way, this differentiation of pericentral and periportal, which is important for this type of analysis, is traditionally done by immunostaining CYP2E1. So the CYP2E1 positive region is pericentral and the negative region periportal. But this cannot be done in intravital imaging. In intravital imaging, we differentiate the periportal and pericentral areas by injection of the mitochondrial dye TMRE. And because this enters the lobule in the periphery, these regions where it is more intensive, this indicates the periportal region, and where it is less intensive, the pericentral regions. And this can be automatically then quantified so that we get the intensities separately for these zones of the liver lobule. Important control also of the intravital imaging. When we see such blebs, it would theoretically be possible that they are an artifact due to the injected fluorophore, here the CLF, the bile salt analog. Therefore, we validate this by immunostaining of tissue which did not receive CLF injections. And bicanaliculi can be immunostained by, for example, CD13. And there we also see these blebs, so they appear independent of the externally administered, exogenously administered bile salt analog. An important further analysis is now the tissue bile acid concentration. And we did this by MALDI-MSI. So here, immunostained tissue is used, and this is a CYP2E1 immunostaining to indicate the pericentral regions of the liver lobule. And on these immunostainings, the MALDI signal is superimposed. So here it's the MALDI signal for taurocholic acid. The disadvantage or a limitation of this method is that MALDI has a relatively low resolution, so we can never see individual cells, but we can differentiate pericentral and periportal regions. The strength of multi-MSI is that the identified chemical is very specific. So the signal we see here, so there we can rely on that this is really taurocolic acid. It's mass spectrometry. And what we see is this transient increase of TCA in the center. These are always the centers of liver locules, the pericentral regions. And, but this is transient, already 4 hours after overdose, it's much lower. This is the quantification showing this transient increase, and that this increase occurs preferentially in the pericentral region. Of course, we confirmed this by conventional mass spectrometry of homogenized tissue. This showed a similar result, also a peak, transient increase about 2 hours after overdose. These results motivated us to look a bit closer at tight junctions proteins. So 1 and Claudin 3 are tight junction proteins of the bicanaliculi. And what we could see is that at 2 hours after overdose, they are not only widened, but give such a fragmented impression. Therefore, we asked the question if these morphological changes have also functional consequences. And what comes now is perhaps the main achievement of this work. So we established 2 procedures to study the tightness or the functionality of the bile-blood barrier in intact livers of living mice. And the first technique is to study leakage from blood to bile. And for this purpose, we used a high molecule fluorophore labeled dextran. So you will see this by green fluorescence. And we begin again with the control mouse. So there you will see the intravenously injected dextran in the sinusoids. And please focus on the bicanaliculi. Here you see this bicanaliculus as the black structure between hepatocytes. If I start now the video, you see the dextran, and you see that the bicanaliculi stay black. So there is, in a healthy situation, no leakage of dextran from blood to bicanaliculi. It's different to what we have seen for CLF, as there is an active transport, but not for such a large molecule within this period. Now we go to the situation very early after APAP intoxication, so 1.5 hours after overdose. And perhaps focus on these indicated regions, perhaps ideally here. So here you see a widened bicanaliculus and the sinusoid comes and it goes in. Perhaps you see it even better in this magnification. So here comes dextran, and after this, it enters the bicanaliculus. Okay. Yeah, here and stop. Now it is in the sinusoid, and a bit later, it leaks into the bicanaliculus. So this functional analysis allows us to conclude that APAP induced leakiness of the bile-blood barrier from blood to bile. And next we studied the opposite direction. Is there leakiness from bile to blood, which is a bit more complicated? For this purpose, we used another fluorophore, CMFDA. So CMFDA passively enters the hepatocyte. CMFDA per se is not fluorescent, but by esterases in the cell, it is cleaved to 5-CMF, and this shows fluorescence, and is also a substrate to BSEP and MRP2, so it is secreted into the bicanaliculum. This gives the opportunity to study if the secreted 5-CMF 10 leaks from the bile canalicus to the blood. This is analytically a bit more challenging, but I think you can see it. So let's focus again on the control situation. Here you see the 5-CMF in the bile canalicus. It's a different situation as before, because you don't see it in the sinusoid, but this is because CMFDA per se is not fluorescent. So you only see it in the hepatocyte, and then especially when it is secreted into the bile canaliculi. And when we quantify green fluorescence in the sinusoid, it always stays close to 0. So this is a background signals. Now the same situation in mice early after APAP overdose. See it in the bile canaliculi, and now there is also some signal in the sinusoid. It's perhaps not so super intuitive, but when we quantify the CLF signal in the sinusoid, there is a clear difference, clear increase compared to the control situation. So this means that we can conclude that there is also leakage from inside to outside, so from the bile canalicus to the sinusoidal blood. Yeah, now let's move on to a next question. We have now repeatedly seen that APAP overdose increases the concentrations of bile acids in the hepatocytes in vivo. But of course, this should not be per se not be overinterpreted. The question of relevance is if this increase goes beyond thresholds of cytotoxicity. So can it really contribute to the hepatotoxicity of paracetamol in vivo? And to study this, we used our cultivated systems. So these are cultivated hepatocytes, which exist either as sandwich cultures or as spheroids. We are always careful with such in vitro systems. There are a lot of differences to the in vivo situation, but some aspects are maintained. For example, the uptake of bile acids by NTCP and OATP and the secretion into bile canaliculi. This is quantitatively a bit different, but in principle it works also in vitro. So what we did is we used bile collected from the extrahepatic bile duct from mice. And we determined the total bile acid concentrations and incubated hepatocytes with increasing concentrations of the bile acids. So this was done for native hepatocytes and also for hepatocytes co-incubated with paracetamol. But let's focus on the native hepatocytes. And so there we can see that the EC10, so the concentration of just beginning cytotoxicity is 98 micromolar, and the EC50, so where 50% of the cells are dead, is 542 micromolar bile acids. And now we know which concentrations show a beginning and a severe cytotoxicity, and we harvested the hepatocytes and analyzed the intracellular bile acid concentrations. And with increasing concentrations in the culture medium, the intracellular concentrations increase. And this could now be compared to the in vivo situation. So here we see again that after APAP overdose in vivo, the tissue concentrations of bile acids increase. And when we isolate hepatocytes from these mice and analyze bile acids and normalize it in a similar way as in the in vitro experiment, we see that APAP causes an increase of intracellular bile acids to approximately 400 micromolar. And these are indeed concentrations which are reached in cultivated hepatocytes exposed to cytotoxic concentrations of bile acids. So this means that the extent by which APAP increases intracellular bile acid concentrations indeed reaches thresholds of cytotoxicity or even transiently exceeds these thresholds. So by having now studied the extent and relevance of these increased intracellular bile acid concentrations, we next ask the questions if we interrupt or if we block the uptake of bile acids, should this then not have an influence on hepatotoxicity? And there were already established methods to do this. So the two carrier systems take up bile acids. These are the NTCPs, or SLC10A1, and the OATPs. By the way, in human, this is almost exclusively the NTCPs, but in mice, both systems take up bile acids. And there are knockout mice available where the OATPs were eliminated. And for NTCP, a clinically approved inhibitor, Mercudex-B, is available. So therefore, we use OATP knockout mouse and studied with and without the Mercudex-B treatment. And this is the situation after paracetamol intoxication in the wild-type mice with these dots, which represent placental necrosis, and already macroscopically, the situation in the OATP knockout plus Mercudex-B mice is quite different. Also in the histology and after quantification, we see that the dead cell area is strongly decreased. Also, the increase in liver enzymes is drastically reduced. This is not the case. One has always to perform control experiments if by OATP knockout or Mercudex-B, the activating enzymes are altered. This is not the case. And also the total glutathione content is not or only to a relatively small amount influenced. So in conclusion, this intervention by blocking bile acid uptake indeed ameliorated paracetamol hepatotoxicity in mice. And but what is interesting and important, this intervention did not ameliorate the compromised bile canaliculi. So this is just unaltered. But of course, also not unexpected, because the classical mechanisms of paracetamol, its activation to NAPK and formation of reactive oxygen species, this is of course not altered by uptake, bile acid uptake inhibition. But what we see is a strong difference, massively reduced bile acids in liver tissue, also confirmed here by mass spectrometry, and vice versa. The intervention causes more bile acids in blood, which is of course clear if they are not taken up by the liver, serum concentrations increase. Yeah, this was the summary of the breach of a blood barrier by paracetamol. And before I finish, I would like to show you that this is not an unusual situation. So the apical hepatocyte membrane is indeed a kind of Achilles heel for many types of liver damage and liver diseases. So we have seen now a scenario where the biocanaliculi leak to the sinusoidal blood, but the membrane itself remains intact. And there are different situations how the apical site can be damaged. And one, the acute scenario is if there is an acute obstruction, for example, experimentally done by bile duct ligation in mice. This is a very well studied mouse model early after bile duct ligation, the concentrations of bile acids massively increase in the biliary tract. And they even increase to so high concentrations that it's a surprise that the liver survives this. I think a mechanism how it does this is that single dispersed hepatocytes die and they release bile acids into the sinusoidal bloodstream. And this decreases concentrations of bile acids in the biliary tract in the acute situation. How this works could also be studied. I will only show the summaries by this type of imaging. What happens is here a rupture of the apical membrane. It's not the tight junctions, it's a real destruction of the membrane. Then the cell dies, also the basolateral membrane becomes leaky. And so the highly concentrated bile leaks into the bloodstream. By the way, that's quite interesting. Of course, one should not humanize livers, but this is a bit egoistic behavior. So the liver protects itself from tissue damage by releasing too high bile acid concentrations into the blood at the expense that this damages other organs. So this leads, for example, to kidney damage then, but only chronically. This is how the liver protects itself against the acute organ failure. And just two videos how this looks. This is now a bicanaliculus filled with CLF, only to see the content of these bicanaliculi. And this is day 1 after bile duct ligation. And you will see how just a second, I hope I will. Yeah, you see how this bicanaliculus will swell and finally rupture. So this is the initial event of something we name bile infarct formation. And you also see that via this ruptured membrane swells, ruptures, and then fluorescent material is periodically released into the bloodstream. This is how and you don't see the canaliculus any longer, so it has emptied its fluorescent material to the bloodstream. But this is only the beginning of something which we name the domino effect of bile infarct formation. Such single dispersed ruptured hepatocytes have a tendency to cause this domino effect so that also neighboring hepatocytes are compromised. And so here you see how these infarcts release fluorescent material to the bloodstream. Now, this is what happens at the very acute situation with the bile canaliculi. And a final example to show you compromised bile canaliculi in a chronic scenario. So a typical chronic scenario of liver damage is fat and calorie-rich, so-called Western diet. And if you feed this to mice for a year, the liver becomes not only steatotic, but also fibrotic, and finally even hepatocellular cancer is formed. So here you see how over the time these white, also these lipid droplets formed. One can also study them intravitally, but I will not go into this in detail today, but focus on what happens to the bile canaliculi. There is vesicular uptake, and the molecular mechanism is not phagocytosis and not receptor-mediated endocytosis, but macropinocytosis, which we established by using macropinocytosis-specific inhibitors. But just to show what happens there, first again the control situation, the CLF, and it is secreted into the bile canaliculi as it should be. And now to the scenario after 24 weeks of this Western diet. What you see here as these large black dots, these are lipid droplets. And now we also inject CLF, and you can see how it enters the sinusoids. It's also secreted to the biocanaliculi. And when the biocanaliculi are filled, you can observe something which is quite striking. There are these vesicles. Perhaps focus on this region here. The vesicle forming, it enters the cytoplasm and now travels around in the hepatocyte. If you show this in enlarged way, you can see that this vesicle forms, it originates from the biocanaliculi. And this is a typical situation in a chronically damaged liver, where we see this vesicular reuptake of toxic bile. And this contributes to liver inflammation and to progression of steatohepatitis. Yeah, I think with this, I have shown you the scenario how APAP leads to leakage of concentrated bile from biocanaliculi into the sinusoidal blood, but that also for other scenarios where liver damage occurs in the acute and the chronic setting, although the mechanisms are different, but it's often the canalicular structure is a kind of Achilles heel where in liver disease the mechanisms go wrong and contributes to either toxicity or disease progression. Finally, I would like to thank some of the people who did the work. Ahmed Galab is an outstanding expert in these intravital imaging techniques, which I have shown to you, and he contributed to photon microscopy. And then, of course, a lot of image analysis, including artificial intelligence of these videos is needed. This was contributed by Stefan Böhme and Adrian Friebel, and you certainly all know Hartmut Jeschke, who we had very valuable discussions about the mechanisms. And via his contacts, it was also possible to obtain the material and blood from patients after APAP overdose. Thank you very much, and I hope it was not too long. I hope I could present some basic but also interesting new aspects, and it would be great if you could now or also later in smaller circles continue discussing DILI and liver damage by canaliculi. Yeah, thank you very much.
Well, thank you very much, Yang. I think it was excellent talk, and the time was perfect. So it was not long enough because I enjoyed a lot. So does any one of you have any questions, or should I start? Well, I will just start with one maybe a stupid question. Should we rename the name of the cell death after this? So is it maybe it's not anymore necroptosis, or is it bile acid induced cell death in not only in IPAP, because you also showed the BDL, no, and FLD.
Yeah.
So what do you think?
Very good question, Shobi. I must admit I cannot yet answer it. We are working on this. So the cell death after paracetamol has of course been studied a lot, and much work comes from, for example, from Hartmut Jeschke's group. And he describes this molecular mechanism which goes via Jun kinase, transfer to the mitochondria. And he says it's neither real apoptosis nor necrosis. It's a kind of programmed necrosis. This is how he names this molecular mechanism. And now with knowing that in vivo overloading with bile acids contributes to the death, we have now to carefully differentiate between studies which were done in vitro with APAP exposure, which of course also kills hepatocytes without the addition of bile acids. So when paracetamol concentrations are high enough, it does not need any bile acids to induce cell death. But in vivo, at these critical concentrations of an overdose, which are not yet lethal, but cause hepatotoxicity, there the mechanism of bile acids is very important. And currently we compare these mechanisms with and without nucleodex A. So where we eliminate the influence of bile acids and compare it to a combined effect of bile acids and paracetamol. And it's not yet advanced enough to make a binding statement if the mechanism of death is different. I hope we can answer it in a year or so.
Yeah, that's great. I mean, it should be, no? Because when in the last experiments, when you use this inhibitor, the CYP2E1 doesn't change, no? What it changes is the transaminases and the cell death, but CYP2E1 seems completely stable. So it might be different mechanisms at the same time occurring, no? Which, but this is very nice. I mean, it's really a novel concept, which is really good. Do we have any questions in the chat?
Yes.
Raúl, please.
Thank you, Jan. Fantastic presentation, very visual. I enjoy a lot, really. My question is not quite clear to me if there is any or maybe any correlation between the bile acid leakage and the clinical impact, not only in the liver, but also, for example, the kidney failure that happens in this situation.
Yeah, also very important. And yeah, we are interested in this question and discussed it a lot and cannot give a good answer. So the background is we are a theoretical lab with mice and cells and imaging techniques and no clinic. Yeah, and of course, we are very interested in the translational relevance, and we obtained now this time series of a patient. But this is too little to say, let's say, if there is a stronger initial increase in bile acids, it's prognostically bad. Of course, we discuss such hypothesis, but we don't know an answer. It is also, so we should consider that it's only a fraction of the bile acid which leaks out of the or out of the liver vein. So it leaks to the tissue space, and from there, the largest fraction is carried back into the hepatocytes again by NTCPs and OTPs. So if this fraction, which then leaks through the pores of the endothelial cells through the sinusoidal bloodstream, if this is of prognostic relevance, would be interesting, but we don't know it. And what we are always interested in, if you have clinical connections where such time series in blood are possible, or even human liver tissue, which we could use, for example, for the MSI imaging to see. So this could be even more interesting if one had liver tissue, which is of course very difficult after APAP overdose. But if it would be available for multi-imaging, this could be very interesting, but we are still looking around and trying to find partners who would have such interesting material.
Thank you very much. Very interesting. Thank you.
Jose Juan.
Okay, Jan, congratulations. This was a very nice presentation, very exciting results. And I have a couple of questions for you. The first one is related to ones already asked by Raúl. And if we block the bile acid uptake, we reach concentration close to serum concentration, close to millimolar. I wonder, and I think you have already considered that in one of your slides, that we can expect some extra hepatic toxicity, kidney toxicity, toxicity on the nervous system, or even cardiotoxicity. So this is one of the questions I wanted to ask. And the second one is related to the change of expression of bile acid carriers, both uptake and efflux, when you induce these pathological models. So that can influence the dynamic of this fluorescent bile acid derivative.
Yeah. So to the first question, indeed, blocking the uptake of bile acids by μ leads to a severe increase. But this increase is transient. So for example, in mice, after bile duct ligation, there is an even stronger increase to concentrations between 1 and 2 millimolar total bile acids. And then it takes between 3 and 7 days until one sees the first changes in the kidney. The first symptoms in the kidney are dispersed necrosis of tubular epithelial cells. And we have not yet studied this systematically after such a transient strong increase. There is some hope that this transient increase is not really damaging the kidney, but before such a therapy with μ would be used in APAP intoxicated patients, this has to be, of course, much more carefully studied. But there is some hope that it's too transient to cause severe damage in other organs. Yeah, to the second question. Sorry, Jose, help me again.
The second question is, when you study the dynamic of this fluorescent bile acid derivative, you in principle consider that at the time of your studies, the expression of NTCP mainly and BSEP has not changed, which is not probably true.
Yeah. So in the acute scenario, so within 2 hours after paracetamol intoxication, there such changes are not of relevance. That's too short. But you see the changes in the 2 mouse models I showed in the end, bile duct ligation and Western diet. And yeah, there are 2 systematic changes which we always see, and they are always relevant. So in each cholestatic scenario, the NTCPs are downregulated, and export carriers at the basolateral membrane are upregulated. So this leads to a situation where in a chronic scenario, the bile acids, which are also synthesized in the hepatocyte, do not go to, or do much less go to the canaliculi, and instead exit the hepatocyte to the blood side. So this is the main consequence we observe in the chronic situation.
Okay, because the MRP4 is upregulated too.
Yes. That's it, MRP3 and 4. They go up, they are at the basolateral, so at the blood side, and they export the bile acids to the blood.
Thank you very much, Jan.
Any other questions?
Yeah, great presentation. Thank you very much. So, Guido Stiernimann, I'm from Bern, Switzerland. I have 2 more translational questions. So, if you translate the situation to acute alcoholic hepatitis, there we frequently have this severe cholestasis. And the question is a bit, what is the chicken and what is the egg? And if I watch your presentation, then the problem is not the inflammation, but the bile leakage, and the inflammation is the secondary product. However, we treat the inflammation usually, although there are some hints that addressing the tight junction could be helpful, as has been shown in a non-significant way by Gyöngyi-Szabo in one trial. So what is your opinion on that? And maybe the second question is, in DILI, we frequently have a prolonged cholestasis for 2 to 3 months, and then the patient recovers. And do you have an idea what could be the trigger in that switches back to normal? Is it immune driven, or is it just a repair of the tight junction, or what could it be?
Yeah, it was the first question of changes in alcoholic liver disease. Yeah, this is rather a situation where upon repeated death of individual hepatocytes, replacement proliferation is induced, but also the ductular reactions. Are these scenarios which we are talking about severe, so severe that new bile ducts form? So is it a situation where there is ductular reaction, which you mean?
The problem is usually we don't have biopsy at the later stage where you would see this neo formation of bile ducts. I mean, we can see this in many chronic liver diseases, but usually we do a biopsy at the very early stage of the disease, and there it's more a question about inflammation.
Yeah. So certainly the inflammation process is a pathological principle by itself. So for example, when new bile ducts form and invade the liver tissue, this is always associated with severe inflammation. And what we observe is that some of these newly formed ducts are leaky. So bile acids can at certain positions leak out into the interstitium, which probably contributes to the inflammation. But we have no experiments and evidence that this leakage of bile is the primary principle. So I would tend to say what also I think is the existing main opinion, that the continued toxicity to hepatocytes, that this is the driving force of the inflammation in alcoholic liver disease. And perhaps to your second question, which was more difficult, can you help me again, Guido?
So the point is, in DILI, you sometimes have prolonged cholestasis, blunt cholestasis for 2 to 3 months, and then the situation recovers. And the question is, what recovers? Is it a tight junction? Is it the immunologic driver that maintains the problem, or what could it be?
Very interesting question, and I don't have a convincing answer. It would be important and interesting to address this. In our mice, we don't see this scenario so well. So when we administer APAP or other hepatotoxic drugs, or if we administer CCL4 chronically and then interrupt, the mouse always immediately begins to recover. And that this continues so long, and then at some time regenerates, seems to be a bit more a human situation, if I'm not mistaken. And yeah, would be very interesting to know what happens there in the tissue. Would depend on human biopsies and material. I don't have a good answer.
Okay, Ramiro, go ahead, please.
Yes, thank you, Jan. Thank you very much for the presentation. It's a pleasure always to learn new things. I have just a question regarding the Raul's question. Indeed, I mean, from a clinical point of view, you know that the DILI is classified as hepatocellular, pure hepatocellular, pure cholestatic, or mixed. And what you are showing us today is a typical mixed DILI, let's say. So do you think there are other or many of the drugs which are causing this mixed DILI could also be doing the same? I mean, they may have or may share a common mechanism with this APAP mechanism. This will be one question. And the second one is regarding the type of death, cell death. of the cells. I wonder if the detergent effect of bile acids could be the major, the major effect. I mean, when the bile acids increase so much, could they be doing just detergent effect on membranes?
Yeah, to the first question, we have evidence that yes, it's not only an effect, so this leakage from the bicarbonate liquid blood induced by compromised tight junctions. This is a general mechanism which would be induced by any compound which causes severe glutathione depletion and severe oxidative stress. So for example, if we give BSO to the mice to deplete glutathione, this also leads to widening of the bile canaliculi, but it's per se not enough oxidative stress to cause a similarly severe leakage of bile to the blood. So the tight junctions are not yet so compromised. And we currently study BSO in combination with inducers of oxidative stress. And there is evidence that if there are high levels of oxidative stress, tight junctions always tend to be compromised. Yeah, then to the second question. Help me again.
Yes, the detergent effect is the bile acid detergent effect is feasible at these concentrations.
Yeah, I think so. The killing so the intracellular concentration is in the range of 400 to 600 micromolar, and this is still below a range where bile acids would have a detergent effect.
Okay.
This would be in the millimolar range. And yeah, this rather works via inflammatory mechanisms. The FXR receptor is, for example, overactivated. But I would hesitate to, so it's probably not yet the detergent effect.
Okay, okay, thank you.
Yeah, any other question? Maribel, you want to comment something?
I want to congratulate Jan for this fantastic, really fantastic conference. It's amazing. What we can see now is really incredible. For non-believers, it's just they are fantastic. I was just wondering, because this open new venues, of approaching dealing mechanisms of injury. And as you said, it's not only for intrinsic damage, but also could be for idiosyncratic damage. As bile injury, as I wrote here, precedes hepatocyte death, could we consider that we should explore bile acids? Indeed, we have found, and we have seen increases in bile acids, serum bile acids, as a marker of hepatocyte of drug-induced liver injury. So what do you think about going into that approach for biomarker discovery?
Yeah, so I think when we see early after drug administration bile acid increase, this is of course suspicious and could indicate such a mechanism. What we see here is certainly not, it's not idiosyncratic. It's so a typical concentration dependent toxic effect due to compromised tight junctions, which may perhaps long-term influence immune responses, but not, I would not say after a single dose. Yeah. And it's also difficult to study idiosyncratic responses in mice. They are quite different immune system compared to human. This certainly remains a challenge.
Thank you, Jan.
Yeah, so I'm still thinking, Yang, about the question that the Guido made. I know that you said that obviously there is a lack of human tissue, no, to check what happens to the recovered patient. Did they recover? But have you tested? Because I'm sure that you check the tight junctions in different organs. For example, in the gut-liver crosstalk, when they drink alcohol or something like this, the tight junctions are completely that disrupted, no? So do they recover it? Have you checked this, or do you have any hint?
Yeah. So we did this carefully only in the liver, but it's known from the literature that it happens also in the small intestine, for example.
In the small intestine, exactly. So this could
if the liver survives this initial, so in mice it's 2 to 4 hours, in humans it's much longer. If it survives this initial period, then it regenerates quite well. Then in the surviving hepatocytes, the bicanaliculi are sealed again, so they become relatively rapidly tight.
Okay, that's super interesting.
Yeah, then the clearance is again normal. So that's a relatively short transient period where this leakage occurs. And if the cell survives, it reestablishes efficient tight junctions relatively soon.
Okay. Wow, that's great. That's great. Okay, so if we don't have any other question, do we have any other? I don't see any other. Well, Jan, thanks a lot again for giving us this excellent talk. It's really exciting to have these talks at least once a month, right? and we hope to see you next month, the 28th of April. We will have a young investigator who will present his data that he gathered during an STSM. So this will be on the 20th of April at the same time, 4:30. If you want to join, which I hope you will.
Yeah, thank you.
Thank you very much.
Thank you also from my side. Very interesting, and perhaps there will be some possibilities to interact or exchange results and ideas.
Of course, yeah. Thank you very much for giving us this seminar.
Sure.
Thank you, Jan. Fantastic presentation again. Thank you.
Okay, so thank you to everyone, and we will see you next month then.
Yes, and don't forget.
Take care.
Don't forget the Kaunas meeting, please. We should back them up.
The weekend.
Okay.
Thank you.